So really excited to demonstrate the GloCyte automated cell counter for CSF for you today. This is an instrument that received FDA clearance in 2016 after clinical trials at 5 different sites. So very excited to demonstrate it for you today. Really, when I think back to my time in the lab here at Advanced Instruments, and I think about, why we developed this product, it really came down to 3 things, I would say. One of them was around turnaround time and freeing up tech time. How do we make it so that a tech can juggle the million different things that he or she is tasked with, right, in a given day? Usually, people don't have a ton of free time in the lab these days. Staffing challenges are really rampant. And we as manufacturers look at how do we provide solutions to labs like yours? And GloCyte was one of those answers. So freeing up turnaround time and freeing up or improving turnaround time and freeing up tech time. The other one is around ensuring confidence in your results, right? CSF is a stat test. I remember being at the bench counting, right? I'm juggling a million different things and CSF comes in. I've got to drop everything and I've got to do it. And meanwhile, maybe I've been counting many other samples and I'm pretty tired and there's a lot riding on it because this is an important patient. It's stressful, right? So how do we help mitigate errors that can happen with a very manual method that's used today, the hemocytometer and the microscope? And then tying into that is really the third reason that we developed GloCyte, and that's really around improving staff satisfaction and morale. Again, there's such a burden, I think, on today's techs, and they are doing probably a job that is extremely under-recognized. And it's so important for us to, one, acknowledge the workload they have and make it easier for them so that, and for you, right, so that you can do your jobs in getting those critical results to physicians to treat patients. So that's where GloCyte comes in. So you can probably see right off the bat, and especially as, right, we zoom out and you can see just the footprint of this analyzer. That was one of the things that was really a must-have we heard from labs like yours. Don't give us another huge analyzer. We are running out of space. And so we were able to deliver a cell counter, an automated cell counter for counting cerebral spinal fluid in the footprint of, I was going to say a coffee maker, but to be honest, even smaller. So really you can set it anywhere, almost anywhere in your lab, right? And one of the things I love about GloCyte is how robust it is. And if you were here in the room with me and you could take a peek under the hood of this instrument, what you'd see is that we've really just automated the process that laboratories are using now, that microscope method, except we make it automated. So under the hood, you'd see a microscope objective, you would see a camera, and you would see a light source, because we'll talk about this a bit more. The technology we're using is fluorescence technology. So no moving parts, which for me, that was exciting because coming from the lab, that meant very little to no maintenance. I like that because that means I can maximize uptime and minimize downtime. So we talked about what you would see in the GloCyte instrument. What you won't see is tubing. And I love that because that means there's no carryover, there's no cross-contamination. The results of one patient aren't going to affect the results of another. And we'll talk a little bit more about that. But simple, robust design, small footprint, virtually no maintenance, really simple and straightforward. Sitting next to the GloCyte instrument is the vacuum station, and you'll see exactly how this works. But what this vacuum station does is it will actually facilitate pulling sample through a cartridge that we'll take a look at in just a minute. The sample is contained in that cartridge. None of it goes into the vacuum station itself. And when I talked about virtually no maintenance, I mean it, because truly the only thing that you have to maintain, only every 6 months, is a rubber O-ring in the vacuum that simply gets pulled out with a pair of tweezers and put back in. That's really how simple your maintenance procedure is. So a great thing about GloCyte. If we continue the tour around the instrument, you'll see that we have our sample preparation tray set up here to help us organize our workflow. With this system, we're using fluorescence technology, as I mentioned. So we have our 2 reagents. We have 1 reagent for the white cell test, and we have 1 reagent for the red cell test. The white cell reagent is a nucleic dye. It has a detergent, and the detergent punctures tiny, tiny holes in the outer wall of the membrane, allowing that nucleic dye into the cell to stain the nucleus. Pretty cool. And then on the right, we have the RBC reagent. It's an antibody stain, tags the antigens on the red cell surface. That's really the way that we do this and we make this work. We've got our 2 test cups, one for the white cell test and one for the red cell test. And the piece that I think is so neat about this system is this GloCyte test cartridge. And let me see if I can try to get it in view so you can see it a little bit better. So this is so neat. It has essentially a membrane or filter paper. Right here, there's a white dot, and you'll see it live in action, is we'll put stain sample on this cartridge. We'll vacuum the stain sample through the cartridge into the base, and then we'll trap any cells right on that white membrane. Really kind of magic in my mind, because then this cartridge goes into the instrument. We take a picture of that and automatically count the result and convert it into cells per microliter. You'll see just how fast that is. So really improving turnaround time, increasing confidence in your results, and taking the stress off of techs in your lab. Okay, so 1 cartridge for the white cell test and 1 cartridge for the red cell test. If you look in the lower right-hand corner of the sample preparation tray, we have 2 controls here. One is our low control that contains low levels of both red and white cells. The low and the high levels of QC, we recommend running once daily in compliance with CAP guidelines, along with saline, which is used to make any dilutions, and we'll talk about that in just a minute. And then probably where I cut off is where we were talking about the participation in the CAP proficiency group. The GloCyte is part of the ABF1 survey, which I believe is biannual, so Automated Body Fluids 1. And last time I took a look at that survey, I believe that the N was around 50 instruments, so a pretty robust peer group there. Those controls, once opened, are good for 30 days. They do need to be refrigerated, so we recommend running those at the beginning of the day or the beginning of the shift. The reagents, both the red and white, once open, can actually stay out at room temperature for 2 weeks. So really nice stability there. Because CSF is a stat test, wanted to design those so that there's no waiting time. They're ready to go.